Hi, and welcome to another short video brought to you by Synthego on how you can reach your experimental potential with multiple models. In this day and age, we have so many questions that we want to ask. How do we do a CRISPR experiment? What's the best way of getting high editing efficiency? Are my guides well designed? All of these questions pull us away from the big picture thinking that we generally want to do. What kind of experiment should I run? Do I have enough replicates? Am I using the right model? Can I add an additional cell line? We need to start thinking bigger so that we don't miss the next big thing. Now, one really great story around this is how statins came to life. So statins are drugs that over 200 million people take to lower their cholesterol levels and reduce the incidence of heart disease. Now, how statins were created is a really interesting story on really thinking bigger and not letting that experimental tunnel vision stop us from missing out on critical research. So in the statin story, statins were first discovered by a Japanese scientist, Akira Endo, who identified a compound that could reduce cholesterol synthesis and reduce cholesterol levels, potentially opening up the path to a drug that could lower cholesterol levels and reduce the incidence of heart disease. Now, like any good drug discovery scientist, he tested this drug in mice and found that it actually didn't work. This would normally be the end of the line for any experiment, and we wouldn't know what statins are as we do today. However, Akira, in this case, wanted to think bigger. And so instead of stopping there, he tested another model. And in this case, he tested this in chickens. And because of the biological differences between chickens and rodents, it actually worked. And this really was the pivotal point to develop statin as a drug that is used by so many people around the world. And in fact, this is one of the most successful drugs in the world, bringing in $18.7 billion of revenue in sales every single year, and obviously helping a multitude of people in this case. So how can you leverage this kind of endo-like thinking to really reduce that experimental tunnel vision that we usually get into once we get into the lab? It's always a challenge to try to set up these experiments in multiple cell models like Endo did. But CRISPR allows us to do this relatively quickly, but it has a whole bunch of challenges. How do we optimize for every cell line? How do we set all of that up? And this often limits labs to only using a couple of models or even one model. At Synthego, we can help with this. We have an automated platform that allows us to optimize many cell lines really quickly, and we've already done this for a wide variety of cell lines. So this enables us to help you design and plan those experiments so that you can do them in a broader range of cell lines so that you can see greater insights and greater depth to the biological diversity that's out there and not miss a pivotal phenotype that may lead to the next big thing. As I mentioned at Synthego, we have optimized over 300 immortalized cell lines in a wide variety of tissue types. This image just provides an example of some of the tissue types that we have optimized, and the numbers indicate the number of cell lines that fit into that category. So you can see we have a wide range of different tissue types that are encompassed, which obviously can help expand your work to different models and to different tissue types or the same cell line within multiple for your single experiment. So this really allows you to get limitless cell modeling with our engineered cells services and really reduce your experimental tunnel vision to see much broader and find those insights that we're missing. Thank you for joining us. As always, if you have any questions, please feel free to reach out to the Synthego team at synthego.com/contact.